Hello class, today we'll be talking about the endosymbiotic theory of organelles. How exciting. So what is the endosymbiotic theory of organelles, you may ask? Well, the theory basically states that the early eukaryotic cells, through a symbiotic relationship, took in bacteria such as early mitochondria and early chloroplasts. However, over time, they developed into dependent organelles and were no longer capable of living on their own. The prokaryotes are one with the eukaryote. In 1970, Lynn Margulis proposed a theory to explain the evolution of these two organelles. Margulis argued that they did not develop de novo, or anew, but rather developed from an early symbiotic relationship. Early bacterial species that were able to produce their own energy and were capable of photosynthesis began to reside in early eukaryotic cells. Now, why would these early eukaryotic cells let them stay inside them? Well, these bacteria provided the host with resources, such as food and energy, and in return, the host provided protection from the environment. It's like when a hippie lives on a community farm. Instead of paying for housing or for food, he just works on the farm. Now, over time, this symbiotic relationship became an obligate relationship, and the bacteria were no longer able to live on their own, and developed into organelles. You can see evidence of this by looking at the structures of the mitochondria and the chloroplasts. Now, when we take a look at these organelles, we can see that there are two membranes. We have an inner membrane and an outer membrane. The inner membrane was the original membrane of the original prokaryote, and the outer membrane was the membrane of the eukaryote that engulfed it. So what's another way we can see to make sure that these organelles actually came from symbiosis? Well, the fact that mitochondria and chloroplasts have their own genome reinforced Margulis' hypothesis. Now, these chromosomes are circular, just like the single bacterial chromosome found in prokaryotes. Through genetic analysis of chloroplast genetic material, we can see that it is more closely related to cyanobacteria. And the same thing goes for mitochondria. Their genetic material shows that they are more closely related to proteobacteria. This means that chloroplasts were once free-floating cyanobacteria and mitochondria were free-living proteobacteria. Now, when these organelles became obligate residents, they also transferred some of their genetic material to the host genome. The host then incorporates this bacterial DNA into its own DNA. The term promiscuous DNA has been then coined to refer to this migration from organelles to nucleus. So before we end this discussion on the endosymbiotic theory of organelles, let's sum up some main points. Now, organelles, such as mitochondria and chloroplasts, originated as prokaryotic bacterial cells. They resided in our early ancestral eukaryotic cells. Now, these bacterial cells provide eukaryotes with food and energy in exchange for protection from the environment. Now, after a long time, these bacterial cells become dependent and can no longer live on their own, and so they must reside in the eukaryotic cell and develop into organelles. And lastly, these organelles incorporate some of their DNA into the host genome. So that sums up the endosymbiotic theory of organelles.